We're going to move on now in our discussion of Mendelian genetics. We're going to go past the monohybrid cross to Mendel's dihybrid crosses. Just to quickly review, what did he establish with the monohybrid crosses? He established his law of segregation, right? A parent, when it makes gametes, has two possible forms of any given trait, the traits he was looking at least, that it can pass on to its offspring. Those forms are going to segregate, and each gamete is only going to receive one of those two options. Where is the other one coming from? It's coming from the other parent, right? So the law of segregation he uses now, and he's interested in his dihybrid crosses in following the fate of two different traits instead of just one. So previously he was looking at flower color. Now he's going to say, what happens with if I have two traits? I'm just observing two traits now. And the thing he's trying to answer is, if a parent gives the dominant form of one particular trait to its offspring, does that necessarily mean it also always gives the dominant form of some other trait? So examples of the traits that he's looking at, we're going to follow today in class, are the gene for pea texture. He noticed that he had either smooth or wrinkled peas, and that the smooth condition was dominant over the wrinkled condition. So he gives smooth a big R, and wrinkled a little r. We now refer to these as a gene for pea texture with two alleles, smooth and wrinkled. He also observed pea color. He had either yellow or green peas. And he saw that the yellow condition was dominant over green. So he gives yellow a big Y and green gets a little y. What are the possible gametes that can be produced by a parent that is heterozygous for both traits? These are his F2 plants, right? Remembering from the law of segregation that the parent can only give one of these options to its offspring. So for example, let's say it gives big R to the gamete. That big R can either pair with a big Y from its other trait, or it can pair with a little y from the other trait. If it gives little r can either pair with a big Y or it can pair with a little y. So we're going to do just as we did with the monohybrid cross, except it's a little more complicated now because we're following two traits. So your Punnett square has 16 squares in it now. These are the possible gametes that we just showed. Each parent is heterozygous for each trait, so we're going to write the same gametes along the top that we write along the side. And I'll give you two tips for how to kind of keep your sanity as you start bringing your gametes together to make the zygote, right? One is to leave yourself a little bit of space at the top where you can later go in and write your phenotype. I just indicated it with a red line. The other tip is to keep your R's and Y's together. So instead of writing here RY, RY, keep them like this. Keep your R's with your R's and your Y's with your Y's. The reason for that is because your phenotypes will pop out a lot more readily if you kind of keep your letters organized this way. So we'll pause now, work on your Punnett squares, write your genotypes in the box, and then at the top write in what is the phenotype corresponding to that genotype. All right, here's my completed Punnett square. And here is how I notated my phenotypes. Now you're going to go through, as Mendel did, and you're going to count up your phenotypes. He made thousands and thousands and thousands of trials. And he observed, on average, a certain ratio in the offspring of a dihybrid cross, these F2 plants, right? We've got here, let me get this to go. Okay, 9 peas, offspring that have smooth yellow peas. We have 3 that are smooth and green. 3 that are wrinkled and yellow, and just 1 down here that is wrinkled and green. Homozygous recessive for both traits, right? Here's what those look like. These are the ratios that Mendel saw after thousands of trials on average. He kept seeing this 9 to 3 to 3 to 1 ratio in his offspring. And he used mathematics and the laws of probability to demonstrate that this must reflect independent assortment. And I'm going to show you how he did that. We have a parent that is heterozygous for the wrinkled, smooth versus wrinkled trait, right? And this is just like tossing a coin. If we toss a coin, what is the percent chance that we're going to get heads? It's 50%, right? So let's say we have a 50% chance of getting a big R from that parent to the gamete. We have another parent over here that's also heterozygous for the trait. What is the chance that we're going to get another big R? What is the chance, in other words, that we're going to get big R and big R again? When we use the word and, we're going to use a multiplication symbol. It's a 50% chance again, right? Just like it was before. What is 50% times 50% or 50% of 50%? It's 25%. We have 16 possible combinations in our offspring, right? What's 25% of 16? It's 4. So there should be 4 of our offspring that have a big R genotype. Remember that both parents are heterozygous for Y as well. So what's the possibility, what should be the number, in other words, we said that this is the number that should be big R, big R. What's the number that should be big Y, big Y? It's also going to be 4, right? Because it's the same proportions operating. What is the possibility, what is the percent chance that we're going to get big R, and, remember to use your multiplication symbol, and big Y? We said there was a 25% chance of each. So that's a multiply. And this comes out to be about 6%, which happens to equal 1/16. So we should have 4 that are big R, 4 that are big Y, and 1 out of 16 that is big R, big Y. Let's just walk through our Punnett square really quickly and look at that. Here's my 4 that are big R. I also have 4 that have big Y. And then I have 1 that's big R, big Y. Now, Mendel was not looking at the genotypes of his plants. You cannot just hold up a pea and look at it and say, oh, I see it's big R, right? All you can see is it's smooth. What does that tell you? That tells you that it has at least 1 big R. So now we got to kind of expand our mathematics a little bit here. We're going to add something in. We said the chance of getting a big R from the first parent was 50%. Now we know we're going to have a smooth pea, right? Because all we need is at least 1 big R. And we have to get another allele from the other parent. So there's 50% chance of getting either one. Let's say we focus again on the big R. 50% chance of getting this one and that one. Or, now we use a plus symbol. Or we could have gotten a little r. There was a 50% chance of getting a little r. And that would not affect the phenotypic outcome, right? Because we already have 1 big R. We said this was 25%, plus another 50% is 75%. If we're calculating the same thing for big Y, for the Y allele, what is the percent chance of getting at least 1 big Y? That's also going to be 75%, right? So I denote them like this, R dash, in other words, R plus either one of these other R's, big R plus either one of the other ones, big Y plus any other big Y or little y. So what is the percent chance of getting at least one R and at least one Y? It's 75% of 75%, which is about 56%. 0.5625 times 16 equals 9. So we see 9 that have at least one big R and at least one big Y. This is the exact same thing Mendel saw. These are basically what they look like. Through later study, we were able to realize that these traits that Mendel was looking at were actually on separate chromosomes. And this is the idea that Mendel himself was honing in on. He realized there must be some discrete element that contains that genetic information, or that information that's going to the offspring. And there's some other discrete element that's carrying the information for another trait to the same offspring. And it's just like if you and I both flip a coin, right? The outcome of your coin toss has no bearing on the outcome of mine. So he demonstrates this mathematically. We realize later that it corresponds to how homologous pairs of chromosomes are lining up on the metaphase plate. The chromosome with your P texture information that you got from your mom could line up on the left-hand side of the metaphase plate when the parent is making gametes, or it could line up on the right. And that has no bearing on how the next trait, say for P color, how those homologous pairs are lining up on the metaphase plate. So just a quick summary. 2 alleles for any given trait are going to sort independently from one another. It's not going to have any bearing on any other trait, how they sort. And the offspring are going to display all possible combinations of those traits in particular proportions. And those proportions are what you should see mathematically from 2 independent events. All right, that's the conclusion of my talk on dihybrid cross. But I just wanted to add to the committee that I have a little activity that I came up with. It's kind of a combination of a couple of different activities that I've used in the past, and the students really like it. You use cards to represent independent assortment and random chance draw of alleles, and you use cat coat color genes that for the most part follow Mendelian, classical Mendelian outcomes, but with some interesting variations that you can add in. So the card colors represent the parents, black and red, and then the card suits represent dominant versus recessive in the traits, in the alleles. And then you can follow, and kind of the students can take 6 or 8 genes and figure out, well, what kind of kitten did you make here? So there's like a long hair gene. This is like classical Mendelian behavior. The spotting gene is interesting because it shows incomplete dominance in the heterozygous condition. This one is interesting, the albino gene, because it's actually a temperature-sensitive gene, and you get different density of pigment depending on where in the body the hair is growing. This one is kind of classical Mendelian behavior, the striping, brown or black cat, dense or dilute pigment. Sex-linked red is interesting because we can answer the question, for example, why don't we ever see a male calico cat? Because the X chromosome carries the red coat color, and males only get one. The white gene is epistatic, so it will mask all the other coat color genes. So I just want to share that. It's just an interesting way of showing independent assortment and showing the students that you can get a very different looking cat by altering just one single trait in the outcome. So thank you. Thank you for your attention.